I want to thank the Dravet Syndrome Foundation for inviting me to speak to you today about human stem cell models of epilepsy. Here are my disclosures, not relevant for today's talk. I'm going to talk to you today about two-dimensional human pluripotent stem cell models looking at neuron or brain cell activity using multi-electrode array recordings as well as calcium imaging to try to look for epileptic phenotypes. And then in the second half of the talk, I'll tell you about 3D brain organoid models, so-called mini brains in a dish, to study genetic epilepsies. So when we talk about human pluripotent stem cells, there are two flavors: human embryonic stem cells and induced pluripotent stem cells. And the induced pluripotent stem cell method was pioneered by Shinya Yamanaka, who discovered the technique in 2006, and it was widely recognized as such a powerful approach that he shared the Nobel Prize 6 weeks later. And basically, one starts with a mature cell, such as a skin cell, that gets reprogrammed using transcription factors into stem cells, and then differentiated into brain cells to study epilepsy, comparing control cells with epileptic cells looking for hyperexcitability. These days, we don't have to use skin cells. We mainly use peripheral blood mononuclear cells, so it's a blood draw instead of a skin biopsy to obtain samples. And again, the idea is to compare pluripotent stem cells with an epilepsy gene mutation versus control, differentiate them into neurons, and then do functional assays looking for hyperexcitability with the goal of testing drugs and coming up with precision therapies for genetic epilepsies. How do we characterize the function of human neurons? One way is to do what's called patch clamp recordings, where we put microelectrodes right into the brain cells themselves in a dish. This is low throughput, but a good way to look at firing properties of the cells and different aspects of activity. More higher throughput methods include multi-electrode array recordings, where the cells are cultured directly on plates that have electrodes. And I'm going to talk about that today, as well as calcium imaging. And we have a device called the Incucyte that is a microscope that goes right in the incubator, where we culture the cells to keep them at the right temperature and with the right oxygen and carbon dioxide mix. And without having to take them out, we can image them with the microscope, including looking at calcium imaging, which is a marker of neuronal excitability. So how do we generate the neurons from the induced pluripotent stem cells to study them? One way is to do what's called a small molecule differentiation. And this is taking advantage of what we know about mechanisms of brain development by activating signaling pathways that get the stem cell to make neurons of specific types in the human cortex. Another way is to genetically engineer human pluripotent stem cells to directly make excitatory cortical neurons called iNeurons by inserting transcription factors that drive them toward a specific fate. The advantage of the small molecule differentiation, where you're more faithfully following normal developmental mechanisms, also comes with disadvantages of having very, very heterogeneous cultures that have progenitors, immature neurons, and mature neurons all at the same time. Whereas with the iNeuron approach, you rapidly get all the cells to differentiate into mature excitatory neurons all at the same time. So it's very homogeneous, and they mature very quickly. And the genetic engineering uses a drug, doxycycline, to turn on the program for them to differentiate into neurons. So you time them to get it all at the same time, synchronized. So this is an example of a multi-electrode array approach using what's called the Axion Maestro system, where plates that include 12-well, 48-well, and shown here a 96-well plate all have electrodes embedded in each well, in this case 8 electrodes per well, and the neurons are cultured on the electrodes, and one sees activity. You can look at raster plots that have different action potentials in different wells. You can have heat maps of the activity. On the lower left is an example of wells outlined in red that are from an SCN8A developmental and epileptic encephalopathy patient versus control, where the wells are outlined in blue. These are still immature neurons, but you can see more activity in the red wells from the patient neurons compared to the control. So we studied SCN8A, and when we get the neurons to mature, we see increased activity, and on the left top and bottom are from two controls, and the right top and bottom are patient-derived neurons from two patients with SCN8A developmental and epileptic encephalopathy. And the plots show instantaneous firing frequency over time, and you can see the firing is much higher in the patient cells than the controls. This is shown in raster plots where the pink outlines bursting activity of different neurons in the wells, and you can see much more bursting in the patient compared to the control. And there are different measures we can look at for excitability, such as firing rate, burst duration. One of the useful ones is percent of spikes in network bursts. So these are different neurons within the same well all firing at the same time, and how many spikes in a burst fire. And you can see over time it increases, but it's higher in the patients compared to the controls. And we use this platform to test drugs on the MEA. And for SCN8A developmental and epileptic encephalopathy, phenytoin is the most commonly prescribed drug. High doses work, and we show here without drug at 0 compared to the control, the patient has more spiking in network bursts, but with increasing concentrations up to 80 micromolar of phenytoin, the patient level of spiking is normalized and is equivalent to the control. But that's an existing drug to validate the model, but we want to use it to identify new drugs. And one drug we were interested in is riluzole, which is an FDA-approved drug for amyotrophic lateral sclerosis, or Lou Gehrig's disease, but it hasn't been used in epilepsy. In cell culture, riluzole is known to inhibit persistent and resurgent sodium current, and those are exactly the abnormalities we found in the SCN8A patient-derived neurons when we collaborated with Lori Isom to do patch clamp recordings. So we tested riluzole on the patient-derived neurons similar to phenytoin, and we found that low dose 3 micromolar was able to decrease activity. It hasn't been used in epilepsy patients, but the pediatric epileptologist for 3 patients, 2 of which we had cells to study in our experiments. One patient showed a 50% seizure reduction. A second patient had a dramatic reduction from multiple seizures per day to months of seizure freedom. And a third patient had a dramatic seizure reduction followed by a honeymoon effect, so where the seizures came back. So this is just preliminary data, but it suggests that this approach is useful for identifying precision therapies. I want to move on to calcium imaging. And that device that I mentioned before, the Incucyte, which goes right in the incubator, we use what's called a fluorescent calcium indicator protein, RCAMP. We express it using a virus in the stem cell-derived neurons. And when brain cells are active, calcium goes inside the cells, and this increases the fluorescence when the cells express a calcium indicator. So the timeline for these experiments, again, we use those excitatory cortical eye neurons derived from human pluripotent stem cells. Basically, we plate the stem cells, we add rat astrocytes that increase activity of the cells. We add the virus with the calcium indicator and give the doxycycline to induce the cells to become excitatory cortical neurons. And then beginning day 10, going for 42 days, we scan them every day. This is what the progenitor cells look like 2 days after doxycycline. By 8 days, they're forming nice neurons with beautiful processes. So what the data look like for this calcium imaging is you see, it looks like twinkling stars, and these are increases in the fluorescence when the cells are activated. but you can see at 20 days after doxycycline, so they're not quite fully mature, you see asynchronous activity of different neurons. I'm going to show you a week later. If anyone's photosensitive, this is going to have a strobe light effect, and you might want to look away for the next slide. I'll tell you when it's done. But a week later, you can see all the cells now fire synchronously in networks. So they're mature and they're connected and they're all firing together. And okay, so the strobe light effect is off. Now adding to that, we're using CRISPR gene editing, which many of you have heard of. It's a powerful technique to manipulate the genome, and we can use it to make knockout cell lines to knock out epilepsy genes where loss of function causes the epilepsy, or we can put in specific mutations to make a virtual patient or an isogenic control where the genome differs only by the presence and absence of the one mutation. So we did this for a potassium channel developmental in epileptic encephalopathy. The channel is KCNA2, and we use CRISPR to put in the mutation on a control line, so it's an isogenic control. And at 28 days in the eye neurons, the control line fires slowly and regularly, again in synchronous activity. And when we look at the epileptic mutant line, we can see it's firing much more rapidly and irregularly. So evidence of hyperexcitability where we just put a single mutation in a control pluripotent stem cell line. We've also applied this to Dravet syndrome. So Louis Dang, a pediatric epileptologist in the lab, made a knockout, heterozygous knockout cell line where he knocked out one copy of SCN1A to model Dravet syndrome haploinsufficiency. And we published this line in our studies of SUDEP, but Louis also generated eye neurons, excitatory cortical eye neurons for the Dravet syndrome line and an isogenic control line, and then subjected it to calcium imaging in the Incucyte. I'm not going to show you the fluorescence, but we quantified the activity over time, and this is days after doxycycline, and by 30 days, the neurons have matured in both groups, and over 90% have correlated activity or synchronous discharging. But you can see that the Dravet syndrome model line becomes synchronous sooner than the isogenic control, suggesting the activity is occurring more earlier in development in this line. Now, we also measured bursting activity, and they both start out with normal bursting activity. The control line, as it matures, has lowered bursting activity, but the Dravet syndrome SCN1A het line continues to have high bursting activity, suggesting hyperexcitability. So I think both the MEA recordings and the calcium imaging are exciting tools to look at function in these 2D cultures. So I hope I've shown you that functional excitatory cortical eye neurons can be generated by forced neurogenin induction in induced pluripotent stem cells. The MEA recordings are useful for identifying epileptic hyperexcitability and testing precision therapies. And the CRISPR gene editing is a powerful tool for generating either virtual patients or isogenic controls. And we use the Incucyte Calcium imaging to show hyperexcitability of KCNA2 developmental and epileptic encephalopathy and Dravet syndrome eye neuron models. The next step for the Dravet syndrome model is adding interneurons to the cultures so that you have excitatory neurons, interneurons, and astrocytes all together to more faithfully model what's in the normal human brain. And for making inhibitory neurons, we can also use that forced transcription factor expression to make what's called IGABA neurons or IGNs. And then the goal is then to test drugs to come up for precision with precision therapies for Dravet syndrome. For the next half of the talk, I'm going to shift gears and tell you about human brain organoids. So there are two main ways to make human brain organoids from human pluripotent stem cells. So typically, all protocols start with 3D embryoid bodies, which are aggregates of the stem cells. And then you can have an unguided differentiation, which is you don't treat the cells, you just let them differentiate, and the normal default, for reasons we don't understand, is to make a forebrain. And so you get these complex brain or cerebral organoids that make different brain structures. The problem with this, it's very inconsistent in terms of what structures you make from time to time and batch to batch. So most people have gone to what's called a guided differentiation with small molecules to make brain region specific organoids. So you can make excitatory cortex, you can make thalamus, hypothalamus, cerebellum. One exciting approach is to make what's called assembloids. You guide the differentiation of different brain regions and then you fuse them together to connect them, for example, making cortex and thalamus, and then you can study both regions as they connect together, corticothalamic and thalamocortical activity in the organoids. This is what one protocol looks like. Again, you grow the embryoid bodies in suspension, usually in a spinning bioreactor, such as this, to get nutrients and diffusion of oxygen into the organoids. And they make neuroectoderm and then more complex cerebral tissue. So we used the brain organoid approach first to study a genetic epilepsy that should have obvious structural abnormalities. The class of disorder is called an mTORopathy. So the mTOR pathway is an important regulator of cell function. And many malformations of cortical development are caused by mutations in this pathway. And we're studying a disease called polyhydramnios megalencephaly and symptomatic epilepsy, or PMSE syndrome. And this is an mTOR disorder caused by a homozygous deletion in the STRAT alpha gene that acts on the mTOR pathway, similar to tuberous sclerosis complex. These kids develop macrocephaly, big heads, megalencephaly, big brains. They have severe epilepsy, cognitive deficits, and also areas of focal cortical dysplasia. And Peter Crino showed years ago that these kids have evidence in their brains of mTOR hyperactivity using the marker phospho-S6 that has increased expression when there's mTOR hyperactivity. So we wanted to model this using an organoid approach called SpinOmega that uses this mini bioreactor. And C.C. Plummer, a former grad student in the lab, came up, developed the approach in the lab, and Louis Dang applied it to PMSE. We were excited right from the beginning because the patient organoids were bigger than the control, suggesting it models megalencephaly, or big brain. And first, Louis looked for markers of mTOR hyperactivity, including phospho-S6. And as shown here, the patient organoids in the first 2 weeks showed marked phospho-S6 increases compared to the control organoid. And when he looked at the size, they were much bigger from the PMSE patients than the controls, so it modeled megalencephaly. We found some other abnormalities that you can read about in the developmental neurobiology paper, but we're really excited to find balloon cells, which are characteristic of a specific type of focal cortical dysplasia, but they're hard to model in rodent models of focal cortical dysplasia. But by day 113, we saw balloon cells that express markers of neurons, astrocytes, and progenitors all at the same time that look just like balloon cells resected from human focal cortical dysplasia tissue, including some that were binucleated, which is classic for human FCD type 2B. So we think this model, this organoid model, is very useful for studying genetic epilepsies with obvious brain structural abnormalities. How about functional abnormalities? So for this, we turn again to the assembloid approach that I mentioned. So it's important studying epilepsy to have both excitatory and inhibitory neurons, as I alluded to earlier, but they're generated in different areas of the developing cortex. So this schematic is the developing rodent brain. The excitatory neurons are generated in the dorsal cortex, but the inhibitory interneurons come from the ventral cortex, and they migrate tangentially to join up with the excitatory neurons. So to be able to model that, one can pattern organoids to excitatory cortex, and then pattern other organoids to inhibitory ganglionic eminence, where the interneurons come from, and then fuse them, and the ganglionic eminence interneurons will migrate into the excitatory cortex to form networks. And this is what I did making this organoid when I was on sabbatical in Ben Novich's lab at UCLA a couple of years ago. I generated the ganglionic eminence and the cortex separately. I labeled the ganglionic eminence with a red fluorescent marker 40 days earlier, then fused them, and all these interneuron progenitors migrated into the excitatory cortex to form networks. And what I really liked about what the Novich lab was doing in collaboration with Istvan Modi is they were recording from whole organoids using microelectrodes. It's analogous to putting depth electrodes in a brain. And in doing this, you get really nice oscillatory activity. So this is a control line at about 100 days. You get these oscillations in the normal frequency range, but you don't get oscillations in the putatively pathologic high frequency range between 200 and 400 hertz. When they compared this with a Rett syndrome line, which is a severe childhood epilepsy, instead of the normal oscillations, there were spike-like discharges in the organoids and these high-frequency putatively pathologic oscillations. So the fusion organoid approach to make assembloids is really useful for studying function in developmental epilepsies. Finally, I want to talk about a new technique that we developed in the lab. A talented research investigator, Andrew Tidball, in my lab, reasoned that most organoid protocols start from a 3D structure to make a 3D structure, and they have multiple neural rosettes. And the neural rosette is analogous to the neural tube. But many of you know in normal development of the nervous system in humans, we start with a two-dimensional structure, a neural plate that folds to form a neural tube. So to better model this, Andrew, instead of starting with a 3D embryoid body, started with a 2D monolayer that he cut into uniform squares embedded in matrix, and they round up to form these beautiful self-organizing single rosette spheroids or saucers that he then grows in suspension. And they form nice cortical structures. They make neural stem cells in a ventricular zone that express PAC6 and a subventricular zone that expresses TBR2, the intermediate progenitors. By 5 months, they're making all the different cortical layers. We can pattern them to make ganglionic eminence and inhibitory neurons that express GABA, and we can do the fusion as well. And we applied this to a really interesting disease some of you may have heard, well, many of you have heard of. It's in the past was mistaken for Dravet syndrome, called PCDH19 related epilepsy. It has many different names. It's due to genetic variants in the X-linked protocadherin 19 gene that are loss of function mutations. It causes a developmental and epileptic encephalopathy characterized by clusters of seizures, often provoked by fever. The children have intellectual disability. Later, they develop psychiatric abnormalities in many cases. And interestingly, it affects heterozygous females and mosaic males, but it spares hemizygous males. So these are males that have no normal PCDH19 function. And in most X-linked disorders, males are more severely affected. So why is it in this disease they're not? And the leading hypothesis is something called cellular interference. And the idea is that in females, you have two X chromosomes, and you don't want to express genes from both X chromosomes at the same time. So you undergo what's called random X inactivation. So a given cell will randomly inactivate one of the two X chromosomes. And so a female with a PCDH19 mutation will have some cells that only express the wild type normal gene and some that only express the dysfunctional mutant gene. So when the brain develops, when you have all normal PCDH19 or all mutant, the cells still can develop normally, but it's when you have two populations that they don't interact during brain development, because protocadherin 19 is a cell adhesion molecule that's involved in cell-cell interactions. And when it's present on one cell but not on the other, they can't interact normally. So to model this in human pluripotent stem cells, Wei Nu and Sandra Mojico Perez made lines, knockout lines for PCDH19, and labeled them with red or green fluorescence, and labeled wild type lines with red or green fluorescence, and then mixed them. And the hypothesis is if you mix wild type or knockout and knockout, they'll be normal. But if you mix wild type and knockout, they won't interact. And that's exactly what they found in these 20-day saucers. The wild type with wild type interact normally, the knockout with knockout interact normally, but when you have knockout mixed with wild type, the cells don't interact and you get stripes of red or green cells, but not mixed, suggesting that we can replicate this cellular interference. And we've also, Julie Ziobro in the lab generated a mouse model with, starting with a male that has GFP on the X chromosome with a normal PCDH19 allele, crossing it with a PCDH19 knockout female, so that the offspring are either heterozygous females or wild type females. And in the wild type, the GFP is everywhere, but in the heterozygous knockout female, the normal PCDH19 expressing GFP positive cells don't interact with the knockout cells, just like we found in the saucers. And now we're using both models to test an antisense oligonucleotide therapy. And the idea is to knock out the normal PCDH19 to make it look like a hemizygous male and see if that corrects the abnormality. So just to conclude, I hope that I've shown you that CRISPR gene editing enables the development of virtual patient and isogenic control human pluripotent stem cell lines that we can use to model epilepsy. The 2D human pluripotent stem cell cultures are useful for assaying excitability and testing drugs on patient-derived neurons to develop precision therapies. And finally, brain organoids are a powerful approach for examining genetic epilepsies that affect brain development and neuronal excitability, and again, will be useful in the future for testing precision therapies. So I want to thank you for your attention, and again, thank the Dravet Syndrome Foundation for putting on this conference.
All right. Thank you so much, Dr. Parent, for providing that talk and for joining us now for a Q&A. I've had a few questions roll in, so we'll dig in. The first question was asking, do you think using cells in a dish model or a specific mouse model to specifically mimic the mutation in a patient will be the best way to be able to screen which therapeutics might be the most successful?
So thank you, Veronica. That's a really good question. I think, for many years we've used only mouse models, and it's been difficult to translate into human therapies. So I think the more models, the better. And that's why we're combining the human cell models with mouse models. And I think, as Scott Baraban talked about, zebrafish models have been useful. And I think if you show something works in multiple models, it's got a better chance of working in clinical trials. So that's the direction we're heading.
What do you think about, having multiple lines from individual patients? Do you think that is going to give us do you think that could mimic, why, for example, one patient with Dravet syndrome is successful on a medication like fenfluramine and another is not? Do you think eventually this technology could help us kind of use the cells to trial that instead of using the patients to trial all the medications?
Right. So that's another good question. So it's definitely true with gain of function mutations. For example, in the SCN8A developmental and epileptic encephalopathy, where the mutations can have different effects, different mutations can have different effects on the ion channel. And we see that. And so one drug might work in one patient and not in another. I think with Dravet, that's less of an issue because the vast majority of the mutations cause loss of function. And so if you get something to work in that setting on one patient, it's more likely to work on all the rest of them.
Okay. The next question that came in was asking, how important is it to use these isogenic control lines when using patient-derived iPSC lines to study Dravet syndrome? Why use an isogenic control versus just an unaffected individual's cells to compare?
Right. So that's a great question. So there's inherent variability of the human pluripotent stem cell models. Even within a patient, different lines may act differently. And so between different controls, there's a lot of variability. And so anything you can do to decrease the variability will be helpful. And some of that variability is due to genetic background, similar to what Jennifer Carney was talking about. So by having an isogenic control that only differs in the presence or absence of mutation, you're at least reducing the variability related to the genetic background. And so you can use less lines and have more efficient experiments.
Great. I think another question that I hear a lot, and I think is relevant, especially coming off of Dr. Baraban's talk that we just heard, is, the development of these models seems really great for helping us to better understand disease and choose treatments that might be more effective. But is there any potential in the future for iPSC lines to be used in similar ways to how Scott is doing these transplants? Like, could we take a patient's cells and actually use those to replace faulty neurons in their brain?
Right. So I'm not as negative as Dr. Baraban in terms of the potential use of the human cells. While he's correct that currently we have trouble making some of the MGE interneuron types in culture, particularly the parvalbumin cells that he and Dr. Goldberg talked about, because they're born very late in development, so they're tricky to make in culture. But we're a number of labs are working on ways to do that. So I think it's just a matter of time where we'll be able to make the right cell types that are migratory after transplantation, that become the right types of interneurons. And obviously the other factor, as Dr. Baraban discussed, is we need to take out the tumor risk, but I think that's possible in the future as well. So I think we shouldn't just narrowly go one way or the other way. I think we should try pig cells, but we also need to optimize human cells and try those, because eventually you want to take the patient's own cells and transplant them back into the same person.
Okay. I just got a question through the chat asking if you've looked at mesenchymal stem cells yet as a source.
No. So, adult stem cells are useful for certain things, but for example, mesenchymal stem cells are have positive effects based on their anti-inflammatory properties, their pro growth factor properties, but they don't really become functional neurons to any great extent. So I think they have a limited utility.
Okay. Well, thank you so much, Dr. Parent. I think we are already so behind that we're behind our break. So I think we'll cut the questions off there, but thank you so much for joining us and for giving that really great talk.
Thanks, Veronica. Bye-bye.
Bye.